She says,
hello, I'm Julia Rozanova, and I'm a researcher at the Yale School of Medicine, whose work is at the intersection of HIV, aging, and addiction, and who absolutely cannot record good videos, so please bear with me. I have received continuous NIH funding over the last 6 years, and I owe this continuity of support to some outstanding individual. When over the last year, my colleagues at the university and the NIH staff heard my SOS call for help, they could have very easily said no, but instead they chose to work with me and to think outside the box to find solutions that offered us bridging. And at the end, my story was a happy one because our R01 grant was funded last month. So I think it symbolizes HIV research more generally, to come to the rescue despite all odds. I salute such individuals who can help us all to weather great storms. And I think that by working together, building those bottom-up efforts, we can have a system where NIH, the academia, and clinical and business communities can work to advance HIV science and HIV care. Thank you very much.
wow, hi there, Mitchell.
Hey, Judy, great to see you.
Partner in crime, another one of the very, very important initiators and planners of this incredibly rewarding experience and live stream that we're having. And I'm going to turn things over to Mitchell, who's going to host a really important panel discussion around networks. We talk a lot about individual researchers, but we also have networks of people. So I'm just going to hand it right over to you, Mitchell. Carry on.
Great. Thank you so much, Judy. And I'm Mitchell Warren, the Executive Director of AVAC. And welcome to hour 3 of this remarkable marathon. If you missed hours 1 and 2, don't worry. All of this is going to be uploaded and archived on the saveaidsresearch.org website. So it's all going to be there, not to worry. But you have missed 2 terrific hours. And I'm really excited for this 3rd hour, where we're going to focus on 4 clinical trial networks that are funded through the NIH and work on a range of issues, which you'll hear about in the next few minutes. I did just want to say that last video you saw and the other testimonials, they will also all live online on saveaidsresearch.org, because saving AIDS research is not a 24-hour exercise. It is a commitment from all of us involved. So if you have a story to share, please go to saveaidsresearch.org, and you can see in the navigation about sharing your story. And everybody has a story, because as you heard in the first hour, HIV and AIDS research influences not just the HIV/AIDS response, but it has influenced every aspect of biomedical, social, and behavioral science. And you'll hear more about that in this hour. So again, welcome to hour 3, and I'm going to welcome 4 terrific investigators who lead these 4 clinical trial networks. And for this hour, what we're going to do is hear from each of them in turn about their clinical trial network, what have been the contributions of each of the networks, and what might they discover next if resources continue to flow. So I'm delighted to welcome, in some particular order, the people popping on your screen right now: Larry Corey, Joe Eron, Mike Cohen, and Sharon Nachman. And as I said, they represent hundreds, if not thousands, of investigators around the world studying pretty much every aspect of HIV and AIDS science and beyond, as you'll hear. We're going to do this alphabetically by network. And so we're going to start first with Joe Eron, who's at the University of North Carolina in Chapel Hill. And he is one of the leaders of the ACTG, which has for many decades, and he'll tell us how many, really led discoveries across treatment and other aspects in HIV and AIDS. So Joe, over to you.
Great. Thank you. Mitchell, thanks, first of all, for doing this. You are wonderful, and we really appreciate you and Judy and the whole team. So this is the ACTG. This is our mission. Our mission is to cure HIV infection, reduce the burden of disease associated with HIV and its complications, And we're also really part of, tuberculosis research and viral research, as well as emerging infections. We've had a lot of contributions. We started in 1987, so this is an old network, but a vibrant one, almost 40 years. We've made landmark discoveries in antiretroviral therapy, starting with AZT, but really continually over the last 3.5 decades, and most recently studying long-acting therapy in people that have adherence challenges. We developed effective treatment and prophylaxis for opportunistic infections, and these treatments are now used for many populations of people that have immune compromise, not just people with HIV. Our scientists, clinical scientists, were among the first to recognize the connection between HIV and cardiovascular disease. And recently there was a very large study that we helped run that showed that giving statins to people with HIV who have low cardiovascular risk actually improves survival. We have a long-term cohort with people who have been suppressed on therapy and followed for 15 to 20 years, and we're studying the HIV reservoir in those. We're really a leading network, and I'll show you in a second, in tuberculosis treatment and prevention of active tuberculosis disease. We work on hepatitis B, and as you'll hear from all the other networks, we really contribute to emerging infections when those emerging infections arise. This is just a quick map showing you that we're all over the world. We're in North America, South America, Africa, Southern Asia, and Southeast Asia. And you'll see this is true of everyone who speaks here. We are all over the world. And this network of sites that's made up with all the networks is really fundamental and foundational for HIV research and research on TB and emerging infections. This slide was supposed to be a build, but it's not, but it's okay. On the top, I'm just showing you that this is actually the causes of death in the United States from HIV, starting around the year 1990. And each of these little dots on here, these little are studies done by the ACTG. One in particular, landmark study showing that 3 drugs improve survival. And you can see how the ACTG has kind of contributed to survival and improvement. So, and even just as I mentioned, as recently as a year ago, this study on long-acting therapy for people with adherence challenges. Over here on the bottom left these are all the HIV drugs, and the ones that are highlighted in yellow are ones where the ACTG contributed to the development. We actually our network actually demonstrated that you could give 1 month of prophylactic therapy to people with HIV who also were exposed to tuberculosis and prevent reactivation, as well as 9 months. So 1 month instead of 9 months. And then for the first time in 40 years, the ACTG, along with the CDC, demonstrated that you could shorten tuberculosis therapy to 4 months from 6 months. That's the first treatment shortening in 40 years. So what do we need to do? We're focusing on this. We need to take therapy developments like long-acting therapy and make sure they reach more people, and especially people who are challenged by taking daily therapy. We need to incorporate novel treatments and novel developments into antiretroviral research, because the goal is to achieve virologic suppression for everyone in the US, even people that are hard to reach, everyone in the world, even people that have trouble navigating pills or adhering to medication. We want to develop a scalable therapy for HIV remission and cure. That is one of the major goals of our group, and obviously we need to continue to do that. We want to understand, treat, and prevent the comorbid conditions that are associated with HIV. We want to optimize therapy and prevention for drug-sensitive and drug-resistant tuberculosis. We want to be part of a cure for hepatitis B for people who are living with both HIV and hepatitis B. And we will evaluate emerging therapies for emerging infections like we did with mpox and like we did with COVID. And finally, if we don't go forward, then we'll still have 30,000 infections of HIV in the United States every year. Long-acting therapy, that's a novel approach, won't make it to low and middle income countries. Developing long-acting therapies for both hepatitis C and tuberculosis will be delayed and might not happen at all. The innovative science that we do that has the potential to enhance inflammation, immune dysfunction, and aging, not only for people with HIV, but for all people, won't happen. And the foundation for HIV cure will be left to other countries, and it'll flounder. And essential research expertise and the research foundation infrastructure will be lost. So thank you. I love being part of this group, and I'll turn it over to my colleagues.
Great. Joe, thank you so very much. And we'll come back when we have the panel discussion about many of the issues you raised. But, the very first hour of this program talked about the benefits of AIDS research. And it's not just about HIV and AIDS. And you really beautifully highlighted that. And we'll come back to that as well. Next, we're going to go from the ACTG on the therapeutic side to the prevention part of the response. And I'm delighted to welcome Mike Cohen, also coincidentally enough, based down in North Carolina, to give us an update on the HIV Prevention Trials Network, the HPTN. Over to you, Mike.
Thanks, Mitchell. And I think I see Joe, but not slides. All of Joe's attractive. So the HIV prevention, thanks, Mitchell, for organizing this, and I'll try and be brief and linear. Waf El-Sadr and I direct the eponymous HIV Prevention Trials Network, whose goal it is to prevent HIV. We are really married to, in every possible way, the HIV Vaccine Trials Network. So the way you would look at this is that their goal, and Larry will talk about this, is to make a vaccine, things like a vaccine. Our goal is to do everything else possible short of a vaccine to prevent HIV. And if I control the slides, I'm told, let's see if that's true. Okay. So we started in 1999 under the leadership of Ward Cates. Waf El-Sadr and I have directed the network for about 10 or 11 years. We have enrolled during the course of these years 172,000 participants. We have 78 ongoing or completed trials. We have 69 sites distributed around the world in 13 countries. And now, we are very sensitive to the idea that HIV infections that we're trying to prevent can occur in the general population. And so you'll see more than 100,000 people in the general population have been participants in our studies. But we've also looked at couples, adolescents and young people, cisgender women, MSM, transgender people, pregnant women, and people who inject drugs. We've also recognized, and this is currently important, while we're a global network, and we think, and working globally is really critical in order to accelerate the velocity of discovery. In all candor, I don't think we've made we've made very few of our discoveries without working multinationally. We still have an obligation to work extensively in the United States. We work primarily in the southern United States. We've had more than 10,000 US participants in our studies. I'll go to the next slide. So as Joe did, I'll try and summarize what we think are some of our bigger contributions. I'm not going to go through, you'll be very glad to know, 78 studies. But a landmark study now many years ago was HPTN/HivNet012, in which we were able to show that by treating moms at the time of delivery, we could, with nevirapine, no longer so popular, we were able to prevent the mother-to-child transmission of HIV dramatically. And nowadays, HIV transmission mom to baby in countries with appropriate treatments available is exceedingly rare. In fact, I think there were only 53 such transmissions in the United States last year. So that's a real contribution. We worked for probably more than 20 years, but a decade of trial experience to demonstrate what is a really simple point, that when you treat people successfully, when you offer them the treatments that Joe indicated, and you suppress viremia below 1,000 copies, the transmission it goes away. HIV sexual transmission and most probably all other means of transmission are eliminated. That is sexual and also bloodborne. This study was called HPTN 052. It was identified as the scientific breakthrough of the year. And in this study, we also demonstrated or started to solve a problem that has been resolved, which is that delaying treatment is a really bad idea. So immediate treatment, as soon as diagnosis is made, both eliminates transmission and improves health and ultimately will lead to a near normal or normal lifespan if treatment is started early enough before CD4 count is depleted. Before this study was completed, the HPTN 052 study, and then ultimately by other investigators, the START and Temperano studies, there was a delay in treatment, and that delay was unfortunate and no longer exists. And this HPTN 052 study led to two really important considerations. One is called 95-95-95 by the WHO, which logically tries to identify 95% of all people infected, link that 95% to care, and then ultimately have 95% experience durable viral suppression. Now, in countries that have achieved 95-95-95, where PrEP is not available, where all you have is behavior change and 95-95-95, in countries like Eswatini and Botswana and several others, the incidence of HIV in the general population is like 0.1, 0.2, 0.3%. This has been demonstrated in studies called FIA studies. So we know treatment as prevention works, and the difficulty is making sure that it can be established. The other point about treatment as prevention, also referred to as TASP, is the U=U campaign. When we completed this study, Bruce Richmond, quite an incredible activist, started the U=U campaign, where undetectable is untransmittable, and it galvanized people to get treated and reduced stigma and created better loving relationships. So we're extremely proud of this study. The studies that I haven't highlighted have been important, but I want to use my time to focus on the highlighted studies. What's happened in the last few years is a couple of important things. First, because of the difficulty of daily pills for people who are otherwise healthy, that is oral pre-exposure prophylaxis, long-acting PrEP has become popular. In our HPTN 083 and 084 studies, we were able to develop, in collaboration with ViiV, cabotegravir, which is an important drug both for treatment and prevention. And more recently, in collaboration with Gilead, we worked on lenacapavir. There are 4 Gilead studies. There are now more than 4, but there were 4 Gilead studies, purposes 1 through 4. 1 and 2 with the fundamental studies that showed that lenacapavir could work for at least 6 months. 3 was a study that is enrolled and has demonstrated enthusiasm among women at risk in the United States to take lenacapavir. And 4 is a very successful study of lenacapavir for people who inject drugs. And we continue to collaborate with both of these companies. And as you probably know, the NIH is committed to a very large investment in the implementation of status neutral prevention activities. That means TASP and PrEP, and they're particularly interested in long-acting PrEP. STIs, classical STIs, go along with HIV, and HIV, of course, is an STI. We've participated in a vaccine study for gonorrhea called the Bexsero study, and those results should be out soon and are quite important. We have an ongoing study called HPTN 096, which is to prevent HIV in the southern United States. Much of HIV in the United States is in the southern United States, and this is a community-based trial that we're very excited about. And as Joe said, and I think should not be overlooked, is these networks, people could complain or can complain. The networks are expensive and take resources and essentially compete with individual investigators, but they serve a very different purpose. They're designed both for very large studies, for velocity, and importantly, to create stable infrastructure for catastrophe. So when COVID came along, all the networks on this call, but especially the VTN, PTN, became the COVID prevention network. And we became, Larry Corey and I became COVIDologists overnight. And Larry will probably talk more about the contributions we made in COVID vaccine discovery and in COVID monoclonal antibodies. And as Joe said, the networks are available and do collaborate in multinational studies for the treatment and prevention of emerging pathogens. And that infrastructure, above and beyond all the contributions in HIV, should not go unnoticed. Going forward, we will do more of the same. We'll continue to collaborate with our colleagues on these networks in HIV prevention, what we refer to or think of as status neutral prevention, something for everyone. If you're positive, we have something, and if you're negative, we have something. And we would hope to aggressively continue those activities going forward. So thank you for letting me speak on behalf of the Prevention Trials Network, and I'll turn it back to you, Mitchell.
Great, Mike, thank you. And that is actually a perfect segue as you ended there, talking both about the CoVPN and the HPTN's work on prevention, as you said, up to vaccines. And I'm delighted to go from North Carolina to Seattle and to welcome a great friend and colleague, Larry Corey, who leads the HIV Vaccine Trials Network for a number of years now, and as you heard, also the CoVPN. So, Larry, the floor is yours.
All right. Well, thank you all. Let me, I think, see our slides. Hopefully,
You can use my slides.
You could present. You could always present Mike's. I know you're good collaborators.
Yeah. And I still not seeing them. So
I just saw a flash. Here we go. Beautiful.
All right, we get through that. All right. Well, the HVTN was founded 25 years ago, and its purpose has been to facilitate the development of an HIV vaccine. It came from the recognition that we were not going to cure HIV by antivirals. That latency was established essentially within the first week of acquisition. Now, the network consists of clinical research sites. It also has some very specialized laboratories and also has specialized computational data scientists as we got more sophisticated in our laboratory work. And we evaluate vaccine safety, their immune responses, and in selected studies, there have been efficacy studies. Like all the networks, we are international in scope. I'm a little bit US-centric in the talk today. Later on in the program, Glenda Gray is talking, maybe more in the wee hours in the United States, but she'll emphasize the international sites. But we have 23 US clinical research centers in 13 states. But our sites in South America and Peru and Brazil and our South African sites, as well as our other sub-Saharan sites, are like the ACTG and the HPTN, really critical aspects to our program. Now, we've conducted a lot of studies. We've supported about 28,000 volunteers globally, nearly 10,000 Americans who volunteered their time to achieve the mission of developing an HIV vaccine. We also are part of the networks that are doing trials in TB vaccines and also work closely in long-acting broadly neutralizing antibodies as a novel method of PrEP. And we do that in concert with the HPTN. The studies that we did, the efficacy studies we did with monoclonal antibody VRC01 were conducted in concert with the HPTN. And really they demonstrated that you actually could prevent HIV acquisition by broadly neutralizing antibodies. And that was a key conceptual framework with respect to the issue of turning that around and use structure-based design with those monoclonal antibodies to make immunogens or vaccines that elicit these monoclonal antibodies. And this has occurred since the AMP trial. In animal models, these structural vaccines have been able to achieve neutralization, and we're translating these studies to humans. And this sort of started a couple of years ago, and the early data look like we are starting to do the kinds of sophisticated immunology and sort of train our immune systems to make HIV broadly neutralizing antibodies. And the early data from these, what we call discovery medicine trials, look really good and start to mimicking the antibody responses that one has seen both in mice and in non-human primates. Now, why an HIV vaccine? Well, there's been a recognition politically on the why. Since the global HIV vaccine enterprise was initiated under President George W. Bush, and every administration, whether Republican or Democrat, including President Trump, recognized the importance of investing in the development of an HIV vaccine. And in the US, nearly half of all new cases occur in people who are not recognized at high risk for HIV and would not be identified by self-referral, public health surveillance, or health insurance for injectable oral PrEP. Now, PrEP is effective, but it requires ongoing use, usually for years, and access to a healthcare provider. I would also say, and Glenda will show a slide in her talk, that in areas where generalized epidemics occur, it's really 70% of the cases of acquisition or transmission acquisition pairs who are not identified at high risk. So really, HIV vaccines are essential for cost-effective, long-term, scalable HIV prevention. They can be delivered at lower costs. They have greater accessibility. They can be delivered in pharmacies and in community centers. And really the only proven way to develop the sustained population-based control of an infectious disease like HIV, with asymptomatic acquisition, asymptomatic transmission. And you can't recognize symptomatic cases, something that we recognized maybe a little bit too late in the COVID-19 program. Now, why a network? I think Joe and Mike have alluded to this. I just put it on a slide. A specialized network markedly enhances the success rate of tackling a complicated biologic medical problem that needs iterative human clinical trials. And that's the case with the multi aspects of HIV therapy and cure, which has been iterative over time, TB cure and vaccines over time, and certainly in the area of HIV prevention. It's really the team medicine approach. It builds specialized expertise in laboratory assays and analysis. The specialized labs allow the assessments of different modalities over time. The national and international clinical trials make sure that clinical trial sites enroll into trials as representative of the patient population with the disease. Its unique features are, it's constant over years, it's standardized, and it has transparent decision-making about moving things from early stage discovery to advanced clinical development. And they can be rapidly adapted to new pathogens and problems, as we demonstrated in COVID. As we have established working relationships, we have laboratories that have validated assays, and moving from an HIV neutralization assay to an HIV COVID-19 assay was just part of the DNA of the organization. An HIV research enterprise was essentially, if you like NASA, I like NASA, use that word. It could be the foundation of COVID-19 vaccines and monoclonal antibodies. Now, Americans believe and want an HIV vaccine. We did this exploratory research for our social media campaigns that we use for our registry. This is over 3,000 randomly selected Americans who were tagged on to a polling about voting. So it's essentially voting adults. That's the only skewing here. But a large majority of Americans across political parties believe it's important to develop better preventive methods and treatments of HIV. You can see 74%, and it didn't matter if you labeled yourself a conservative, moderate, or a liberal. And 78% of Americans actually recognize, regardless of their political party, that it's important to fund research on HIV vaccines and monoclonal antibodies. And so there's a recognition of the populace in the United States of the importance of developing an HIV vaccine. Now, it has been a huge scientific challenge. It has more strain diversity than any other virus, and it's unique and being increasingly recognized its diverse immune evasion strategies. Almost all of the immune evasion strategies we now recognize in other pathogens emanated from our knowledge of HIV. It was the HIV vaccine infrastructure that tested and also created the 2SP spike protein that was used in all the vaccines. The HVTN and the HPTN were key components of Operation Warp Speed. And I will say in the last 3 years have brought incredible progress using structural immune design and defining and implementing a strategy for an effective HIV vaccine. And I'll end with the following. There's no question that an HIV vaccine poses vaccine science's greatest challenge. But the scientists funded by the NIH don't give up just because something is hard. That determination has resulted in extraordinary discoveries, bringing us to the brink truly of a roadmap. And I think we really do have a roadmap of truly creating this world-changing breakthrough. And I'll just sort of end on that commentary and point of optimism as it relates to where the HIV vaccine field is. So thank you.
Great, Larry, thank you. And optimism is central to what we all need to be doing, not just this 24-hour marathon, but throughout. I don't think any of us would work in HIV if we weren't optimists on some level. And so thank you for leaving us on that note. Before we go into the panel, we have one more of the networks we want to highlight, and that is the IMPAACT network, which focuses on specific populations that you'll hear about. Delighted to welcome Sharon Nachman from SUNY Stonybrook in New York, who leads that network to round us out, and then we'll go into a conversation.
So thank you very much. It's really nice to be the last speaker because I get to say everyone who spoke before me said exactly what we wanted to say. And Mitchell, thank you very much for hosting it. This is really important. I'm Sharon Nachman. I'm the PI of the IMPAACT Network. Just a brief history. In 1991, just a little bit behind some of the other networks, NIH funded the Pediatric AIDS Clinical Trial Group to focus on these populations that were excluded from research: infants, children, adolescent, pregnant, and postpartum people. And fast forward many decades, and those are the same populations that are often still excluded from research. And it's IMPAACT's mission to continue to study those populations because we need drugs licensed in them. We need to understand how to treat those populations, to prevent HIV, treat HIV, and also get to an HIV cure. And the current structure for the network includes a very strong community presence and leverages collaboration across many groups to make sure that we're doing the best science. What's our mission? Our mission is to improve care for people living with HIV. We have to evaluate novel treatments, interventions for HIV, its complications, as well as TB and other HIV-related conditions. We have many specific aims, and they are specifically related to our scientific committees. They include treatment across the ages, the potential for an HIV ART-free remission, evaluate approaches to TB prevention, treatment, and diagnosis, as well as understand optimal therapeutics and management for neurodevelopmental complications from HIV and its treatment. And each of these are interrelated. You can't have one without the other. And the important part of our network is the collaboration across these scientific aims, these scientific committees, as well as working with the community to understand what do they want solved and how are they going to help us to solve those issues. We have many significant research accomplishments over the last decade. We had so many, I had to put them on 2 slides. And as you can see, I've listed something for every year, because every year we are making strides in the HIV world to make sure that our populations are kept abreast. We're moving ahead, preventing, diagnosing, treating TB, HIV, and other complications. And I want to make sure everyone understands that we're not stopping, that the network must continue because our populations, our community demands it. We have to really understand what is it we can do and how to do it better. I thought I'd show this slide very briefly because this is a list of the drugs that were licensed based on data from our network. And these include drugs that span age groups from birth through pregnancy and postpartum. And they are not just HIV drugs. They are TB drugs. They are drugs to treat COVID. They're drugs to treat hepatitis C. Because again, if we're not going to study them, who will? And what's really important to recognize is that amongst all of these drugs that got licensed through our network, about 70% of the patients that enrolled on these studies were from international sites. So when someone says, can't you just do these studies in the US? The answer is we could, but it would take decades to get enough patients with enough data to get it licensed. And we've already seen that experiment fail, doing a study just in the US. And therefore, our network is international, just like HVTN, HPTN, and ACTG, because we are treating populations with these diseases around the world. They're contributing to our studies. contributing to licensure. So that means that people here in the US are getting access to these licensed drugs because of our international populations. It's not just about treating HIV or treating TB or even hepatitis C. It's also about HIV remission. And I show this slide to remind people that we are moving ahead with our cure agenda, and that 4 of the 6 children who were enrolled on this very high impact critical study have remained free of detectable HIV for more than 1 year after their antiretrovirals were stopped. They are experiencing HIV remission, and we're continuing to enroll infants at birth on this study, both domestically and internationally, so that we can get them to HIV remission and to an HIV cure. Looking ahead, we have many studies that are rolling out, and they're not just for one topic. We're looking at TB studies. We're looking at studies looking at BNABs that can be effective and safe for use in pediatric populations. We're looking at studies that can prevent hepatitis C during pregnancy being transmitted from mother to baby, as well as looking at developing better drugs to treat syphilis. As everyone knows, there is an ongoing syphilis epidemic around the world. The drugs that we have are quite limited, and we need better drugs. And learning from using current drugs and other long-acting medications during pregnancy, we hope to build better medications to treat syphilis during pregnancy so that we won't see any more congenital syphilis here as well as in the US. And these are just a few of the studies. We're also looking at access to new HIV and treatment regimens, both here as well as in the US, because what we have is never enough. And we have to learn to do more with what we have and learn to use new drugs. So I want to say, in essence, thank you to the speakers that came earlier today. Thank you to the site staff, communities, and all of the individuals and families who are engaged in research, because you are what makes it possible. And it is really important to continue this mission if we ever want to treat and get rid of HIV worldwide. Thank you.
Fantastic. Sharon and Larry and Mike and Joe, thank you for setting the stage. I do want to highlight, and everyone can look at the saveaidsresearch.org website to see the program. Many of the scientific topics you heard, from HIV and TB to COVID to international research to vaccines, are going to be covered in more detail throughout the next, I guess I do my 21 hours that we have left in this marathon. But you all provided a fantastic overview of how the networks are structured. And I wonder, I know, Larry, you mentioned, I think, 28,000 trial participants over time. Is there any quantification? How many investigators do you all, represent sitting here on stage, the 4 of you, across the networks? And someone asked a great question that came out of the chat about the interdisciplinary nature of it. We think of clinical trials, but I wonder if you could comment on how vast is this research enterprise, and how, in terms of the number of investigators around the world, but also the diversity of those investigators in terms of the disciplines they represent. Anyone want to start with that?
I would make one comment really quickly. What was amazing to me, and Larry will remember this, when we became COVIDologists, everybody on this call became a COVIDologist, and we had a call every Monday. And that call represented the community you're talking. There were usually between 600 to 1,000 investigators on that call. Is that right, Larry?
Yeah. And that was just the investigators. I think most clinical trial sites internationally have about 20 people per site. And in the United States, maybe about 12 or 13. And even in HIV efficacy studies, we have 40 or 50 clinical trial sites. And in COVID, of course, we had 123. Each trial had about 110. Each vaccine trial had about 110 separate sites. So the skill set of, I think in the last hour of having all the community members with respect to recruiting into them, outreach members, the nursing staff, just the medical aides associated with research. It is truly a team medicine and an enterprise.
Mm.
Yeah, Mitchell, I think one thing that's really important too is that also the diversity of investigators, right? There are people that are studying behavior and adherence, but there are also people that are studying, the genetics and the genomics and the phylogeny of the viruses and the immunology of the responses. Pharmaceutical companies, develop drugs, but in IMPAACT, ACTG, and the other networks, we also collect all kinds of samples to try to really understand how things work. And a lot of our studies haven't just published one manuscript, but there are 20 manuscripts from one study looking at all the various ways you can look at the science, from the smallest molecular piece to the behavior to the epidemiology. So it really is a very broad scientific community. And basically we all get along and we all work together, which we did during COVID. And we have shared studies, as Mike and Sharon pointed out, and Larry and Mike are really have created a tremendous team. So yeah, I agree with you. It really is a remarkable community.
And I just wanted to add that it's not just adult and pediatric care providers. Traditional care providers, perhaps on the OB side or neuropsychologists, are also critical parts of our networks, because it's not enough to have a drug. We need to understand who's using it, how are they using it, are they taking it, or do they not even want to take that medication? And these are incredibly busy care providers. Our OB staff, a shout out to them. They are wildly busy, and I want to say oftentimes the last thing they want to do is get on a study and been on weekly calls and help enroll. But that's exactly what's happening across the networks. Multiple specialties are involved in each of the development of these studies, and then how to complete them, and then the papers associated with them.
That's a fantastic point. And I did want to pick up on a couple of things that you all brought up, but I want to go back to Joe, one of your points. You all, the networks are funded by the NIH, and that's the major, financial input into this. But each of you kind of mentioned collaborations, and I'm wondering, particularly thinking about the pharmaceutical industry and how each of you work with pharma, and as resources potentially decline from the NIH, I think there are some who would say, "Oh, well, pharma can take care of it," and I don't believe that to be the case. But I'd love to hear how each of you both collaborate with pharma and what is gone if the NIH doesn't invest in this research through these networks, if we think it's just up to the pharmaceutical industry.
Yeah, I'll be really quick. The underlying science that really explains the outcome would be lost. I think almost a lot of what we know about resistance to drugs would be lost, because those take secondary and sometimes tertiary analyses. Pharma is unequivocally our partner, and they've been tremendous partners, because they do develop the drugs, and we figure out how to understand how to use them. But I'll defer to others to go more deeply.
I mean, I think HIV vaccines is a different ecosystem here. We recognized, starting with the global enterprise back in 2000, with the Gates Foundation and the NIH, that pharma was not going to be able to do this, that this was a public sector issue, that we are no longer working really hardly with any pharmaceutical companies. And I just had a long discussion with the CSO, CMO of J&J, yesterday about this issue, that needing and wanting NIH and philanthropy to develop an HIV vaccine, that they've done the issue that there is money to be made and they would pick it up and do it. But it requires being shown how to do it. And the resources to actually learn how to develop your immune system and generate B cells that normally are deleted, and to make these unusual mutations, is really something that discovery medicine needs to do. And the NIH has always been the engine of discovery medicine. That there will be a commercial venture there for both monoclonals and vaccines, but the de-risking at this point in time with this field, which is eventually needed for population control. No one, at $28,000 a year for our own country, number of people that would have to be put on for PrEP. No vaccine is going to be $28,000 a year, let alone needing it every year. So that's where the discovery medicine aspects of NIH's research agenda needs to continue to persist.
Fantastic. I do want to just highlight for all the viewers, at 5 a.m. Eastern time, there is a session looking at lenacapavir, the drug that Larry's referring to, that was developed by a pharmaceutical company, but the basic science was funded by the NIH at the University of Utah. And we have Wes Sundquist on at 5 a.m. And he's going to be paired with Linda Gale Becker, who led the trials that got lenacapavir in South Africa, the data. And I think, Sharon, you highlighted how important that international research is. And the third person on the panel is a fantastic advocate, Yvette Raphael, to connect. And so I want to come back to all of you thinking about Yvette talking about lenacapavir with an investigator and a basic scientist. You've talked about laboratories, the clinical trial infrastructure, the essential roles that they play. I also know that each of your networks are deeply committed to ensuring that communities very much are at the center, I hope, of everything we do. And I wonder if each of you could really briefly maybe give a little insight into where science and community meet within each of your networks.
So I can take the hit on this one first. And the answer is straightforward from 0 to 100%. There is no concept sheet that gets approved by the network. No protocol that ever gets developed goes out to a site or enrolls without a community representative being on that concept sheet, on that protocol, at the sites from the get-go. So there's nothing that we do that does not include community from start time 0.
I would just, it's funny because one of the most amazing experiences I ever had to deal with the community. And that is when we did 052, we had 13 sites in 9 countries, each had a community advisory board. But, and obviously there were 1,300 couples as participants, but what I didn't know when the results were known and the results were successful, study was very successful in showing that these couples did not experience transmission. They each had gigantic parties, and I didn't know about this till later. There was a NPR thing about the parties they had, and it kind of drove home to me that this research, which I thought was about me, was really about them. I thought it was all about me. It was really about the participants and the communities who really owned the results of the study.
Yeah.
Yeah, the ACTG is like the IMPAACT, as Sharon. I mean, we have a community member on every protocol. They're on our scientific agenda steering committee. They're on our executive committee. We try to follow that guiding. Nothing about us without us, and we try to do that, and we hope we're hearing their voices, and also that we're disseminating our results. And this is a great example of dissemination to the community, something that's open and accessible. Mitchell, so you should take a deep bow, and Judy and others that are doing this, because the community has access to this, and it's great.
That's fantastic. I wonder if one of you, some questions in the chat rightly about, it's not the full network, but a fifth kind of arm of this investment from the NIH is HANC, the HIV/AIDS Network Coordination Center based at the Fred Hutch. I don't know, Larry, you also at the Hutch. If one of you wants to describe the role of HANC and how it augments and complements the work of the clinical trial networks.
Well, it just does that. We all get together on the phone calls. It sort of bonds us sort of monthly on the community issues and raising points, helping structure even the data that's collected on trials between the networks, improved our sharing of data among the networks, our ancillary activities to the outreach, both to the behavioral science community as well as the laboratory science community and the clinical trials to make data from our trials accessible, how to put it up on websites, how to create a transparent space for it to be seen, and for us as a series of integrated networks to work together with community and do what Sharon and Joe and Mike have said, that it's built into our DNA, from the first concept sheet to the governing priorities and public policy issues that the networks make. It's community that guides us, and it's community actually that leads us.
It's fantastic. And I know, these networks that have been so long established, it wasn't always that way. It's been an evolution, and I've been privileged to be along for the ride and work with all of you. And it really is quite a testament that this is a community broadly that grows and learns. And I want to just really thank Hank, especially, and Russell Campbell, its director. They are one of the, I think now, 40 plus organizations, including the clinical trial networks, that have endorsed this program. And you'll see all of their logos in different parts of the program and on the website, because it truly does take that global village. I did want to pick up on one quick thing, tying back to the last panel. And I don't know how many of you were able to watch a terrific conversation about some of the work, particularly that the Centers for AIDS Research do around mentorship in HIV research. But each of the networks I know also looks at, and I'm not asking this because of who I'm seeing on the screen, what's the pipeline of young investigators in your networks, to complement the experience that is reflected on the screen, and what's the future of the networks, not just scientifically, but in terms of leadership and younger investigators?
Well, I'll just really quickly, I think all the networks we have a scholars program. Ours is primarily devoted. We have a domestic scholars program and an international scholars program. It's really been, and that's been quite important to us. But obviously, in the current last year, everything is up in the air, and the anxiety among investigators worldwide, youthful investigators, is palpable and disturbing. I think the job of people, and I don't want to give the ages of anybody on the call on this webinar, but our job is to try and stabilize the situation, because we're going to lose an entire generation. I mean, the loss, the kind of this exercise we're going through now of Save HIV is so vast in terms of its importance. It's important because of HIV. It's important because of global health, which is kind of built around HIV, all of global health. It's important around infectious disease in general. And probably it's most important in terms of stimulating and inspiring people to become scientists, physicians, epidemiologists, investigators. There's a whole generation that are younger than us that surfaced because of this. And that whole generation is at risk. So our job is incredible cheerleading. Joe and I are in the same university. We have a lot of fellows, and they're very anxious. And we give them chocolate croissants in an attempt to reduce the anxiety, but that's not going to be sufficient.
Well, yeah, I mean, all of us have these programs to bring up the next generation. We recognize that that's both needed, but there's also recognition on their part that HIV is still a discovery medicine field, and that there is room in academia to do this, and there's room, and HIV as a clinical practice has not gone away. And so the issue is how to sustain that. The networks have, we have some of our own programs, and so far we've been able to sustain that. How long that will go in the future, and what the perception will be about the faculty positions. Training goes all the way down to community training and behavioral science training and the recruiters, and there is really a team medicine that's a jobs medicine kind of program in translational research. And this has been an important component of the whole biomedical ecosystem, and HIV really has been the model for that. And will we be able to sustain it, and will be able to achieve the prominence that the United States has had in translational medicine?
I just wanted to add one comment in that the young investigators that we're all mentoring are not just standalone young investigators. They're working with the data from our networks, the samples from the networks, and the statistics and data management groups that we have. So we're teaching them more than just one tone of medicine. We're teaching them statistics. We're teaching them lab medicine. We're teaching them stuff that doesn't necessarily translate, oh, it's HIV or nothing else, or TB and nothing else. But it's much broader, and losing those young investigators means that we're going to have an incredible gap in that next generation of scientific leaders.
Yeah. No, thank you for that, because I think it's, you know, there is, I think, Larry, you described some optimism, rightly so. We need to maintain that. And Mike, there is anxiety out there. And I think that is our collective challenge. And one of the reasons why so many of us came together for these 24 hours to try to balance the anxiety with the sense of optimism and a pathway forward. We are just coming up to the end of hour 3. I want to give you each one last word, literally maybe 2 words. If you can think about, you know, we have kept the viewership super high for all 3 hours. We don't know who all is on. What's the one message you'd want to convey, particularly about AIDS research and clinical trial networks, that you'd want to leave people with? Let me go down on my screen. Mike, why don't you go first?
I think that the HIV research is really quickly has got so many things built on top of it. It's the cement foundation of science of the last 30 years. It's not, it's obviously critically important, everything we've done to change the greatest contribution of our species probably is taking a fatal disease and making it a normal lifespan, you know, for those people who acquire this infection. But it also then served to build this entire universe of success for the United States. So I can't imagine that you're any better use of your time than, quote, save HIV, which is essentially saving all of science. You're really way beyond a single disease in the effort you're expressing.
It's beautiful. Saving HIV science is saving science. Joe, real quickly.
Yeah, I want to say foundational, right? It's foundational for virology, foundational for immunology, the infrastructure of the network itself, the people are foundational and allow us to address whatever it is, another COVID-like outbreak, a flu outbreak, whatever. It is foundational. And I think Mike was saying that, and I think that he's right on.
Great. Foundational. Larry.
What do I say? Major league instead of foundational. I mean, HIV scientists, HIV is at the forefront. The quality of the people in HIV research, the depth of the scientific developments, how HIV is spun off initially, CAR T-cell therapy, the basic immunology, even venturing into areas of aging and cardiovascular disease.
Beautiful.
The people and the quality and where it is led is not just infectious disease. It's certainly the largest depth of all of infectious disease. And its spin-off into both infectious disease research and other areas is just major league.
I love it. Sharon, last word.
I was just going to sum it up and say great science, it benefits everyone.
Love it. Oh my gosh. You had a little bit more time, but that was the perfect summary. I want to thank all 4 of you. Thank you all for being part of this panel. Thank you all for staying online for 3 hours. And we are just about to start hour 4. So Mike, Joe, Larry, and Sharon, thank you all so very much.
Thank you.
Thank you.